video is all about DNA structure, what it looks like, and how it's organized. All cells need some kind of instructions that tell them how to function properly. They need guidelines or rules for making materials in the cells. And in our cells and plant cells and also in bacteria cells, these instructions are called DNA. DNA stands for deoxyribonucleic acid. Deoxyribose is the name of the sugar that is involved in the structure. And nucleic acid is the category of organic compound that it belongs to. DNA is the instructions for our genetic information. Ultimately, the instructions for making proteins, which are our genes. It is found in the nucleus of eukaryotic cells like plant and animal cells, and it is found in the cytosol of prokaryotic cells or bacteria. DNA can be tightly wound and organized into structures called chromosomes. Humans have 46 chromosomes. You got 23 from one parent and you got 23 from another parent, or 23 from an egg and 23 from a sperm, giving you 46 chromosomes or structures, categories, compartments of DNA. DNA structure involves two strands, D double. So here is a strand right here. And then on the opposite side is another strand. And it's twisted into a spiral or a helix shape. So we often call it a double helix, meaning there's two different strands and then it's twisted. Each strand of DNA is made up of chains of units, and each unit is called a nucleotide. A nucleotide has three main parts: a phosphate group, which is located over here, the deoxyribose sugar, which I already mentioned, which is a 5-carbon structure, and then a nitrogen base. The phosphate group and the deoxyribose sugars are the same in every single nucleotide unit along a strand of DNA. However, there are 4 different varieties of nitrogen bases that could be attached to the sugar: adenine, guanine, cytosine, or thymine. So basically, these nucleotides are going to have 4 different variations to them, depending on which nitrogen base is attached at the end. Now, depending on the nitrogen base kind of determines its actual structure. Adenine and guanine are structures that are made of 2 different rings. So you can see there's a ring here and there's a ring here. So the chemical compounds are made of 2 different rings. These are called purines. Cytosine and thymine are made up of only 1-ringed structures, and they are called pyrimidines. One of the easiest ways to keep track of which is pyrimidines has a Y, just like cytosine does and thymine does. Adenine and guanine do not, so they're the purines. So we know that DNA is made of 2 separate strands, and they're twisted together into the spiral shape. But the question is, how do the 2 strands or opposite strands connect to each other and stay connected into this twisted shape? The ticket lies in the nitrogen bases. A scientist named Erwin Chargaff discovered that nitrogen bases are attracted to each other, but not all nitrogen bases will connect with others. Adenines will only connect or form a chemical bond with thymine. Thymine then will only connect with adenine. Guanine and cytosines will only form bonds between each other. So basically, a purine will only bond with the pyrimidine. Now, that means that adenine will never join up with the cytosine, guanine will never show up or join up with the thymine, and the bonds that form in between these are called hydrogen bonds. So if you look here at this diagram, here's one strand of the DNA. Here's the sugar was in blue. Here's where the phosphate groups are. So you can see this kind of makes up the backbone of the DNA strand. So it kind of looks like a ladder, and then the rungs are going to be the nitrogen bases. If there's a cytosine here, then the other strand has to have a nucleotide with a guanine. Here is a thymine, so the other side has to be an adenine. Here is a nucleotide with a cytosine, so this nucleotide on the opposite side has to be a guanine. So A's and T's connect and C's and G's connect. Here's, again, the sugar and the phosphate group. So here's the background. So if you look, you've got this, the two sides of your ladder, which are going to be made of the sugar and the phosphate group. And then the rungs of the ladder will be these nitrogen bases, which will be chemically bonded together. The type of chemical bond that connects the two nitrogen bases, so in this case cytosine and guanine, are called hydrogen bonds. And this is going to be really important when we start looking at how DNA makes a copy of itself and how it's used to make proteins. So, quick review. 2 strands, double-stranded. Here's one strand. Here's one strand. Now, normally it would be in a spiral shape, but we kind of are just showing it in a linear shape right now. Each of the strands is made up of individual units called nucleotides. Each nucleotide is made up of a deoxyribose sugar, a phosphate group, so in this case here's the phosphate group, and then a nitrogen base, either A, T, C, or G. C's and G's will only connect with each other across or forming the rungs of the ladder. A's and T's will only connect with each other. Last thing has to do with how we label each strand of DNA, or the ends of it. The ends of each strand of DNA are either labeled 5' or 3'. And then the opposite end would be the opposite. So if this is the 5' end, this is the 3' end. This is the 3' end, this is the 5' end. And you can tell already that the 5' end is going to be connected to the 3' end. So here's the adenine again, here's the thymine, here's the nucleotide. Okay, the 3' end is going to be opposite of the 5' end. So just out of curiosity, where do we get the 5 and the 3 from? Well, scientists label carbon compounds, which sugar is a carbon compound. They label the different carbons that are involved in it. So here's a carbon, we label it 1, 2, 3, 4, and 5. In this case, the carbon that's labeled 1 is going to be actually attached to the nitrogen base. And then the next carbons are going to be ordered clockwise, just filling in the numbers, 1, 2, 3, 4, 5. So when you look at the ends of the DNA here, you can see that the 5' end, the 5 carbon, or the 5th carbon that would be labeled, is facing the most upright. Okay. In this one, it's the 3 carbon end that is most upright. So we name this part the 3' end and this part the 5' end. And then once that all of the nucleotides are in place, it would be opposite down here. The 3' end is the 3, or the 3rd carbon would be facing downmost. The 5th carbon would be facing downmost here. So that's kind of where the 5' and the 3' end get named. It's based on the number of carbon in the structure, based on how scientists would label those. And this is going to be, again, really important when we start talking about how DNA copies itself and how it is used to make proteins. We'll pay attention to these 3' and 5' ends. So that's DNA structure in a nutshell. If you have any questions, don't hesitate to ask. Have a great day.